In the previous class, we have learnt about fertilization, right? So that is in series we are studying how exactly reproduction happens in human beings. First, we studied about the structures of reproductive system, like male and female reproductive system. Next, we spoke about the mammary gland. Then we spoke about the gamete formation, which is the pre-reproductive phase, where we talk about how exactly gametes are formed in both males and females, that is spermatogenesis and oogenesis. Then we learned about the structure of the sperm. Then we learned about the structure of the egg. Then apart from that, next we started with the process of fertilization in humans. In humans it is actually internal fertilization, and in the just previous thing I explained you how exactly fertilization happens by ejaculation and movement of sperm towards the egg. And after that, egg will fertilize with the egg is getting fertilized by the sperm exactly at the fallopian tube, in the mid of fallopian tube. Then the end product is zygote. The diploid zygote, once it is formed, this is going to develop as an embryo. So we are going to talk now about post-fertilization events. Like I told you, the whole reproduction can be divisible into first pre-fertilization events, fertilization event, then post-fertilization events. So pre-fertilization I explained about the gametogenesis, and exactly fertilization we finished. Now I will be explaining about the fertilization after that, what happens, that is the post-fertilization changes. Here what happens is gametes, when they are fertilized, they form diploid zygote. Zygote will begin to grow or divide and form an embryo. This stage we call it the developmental biology, or we call it as embryogenesis or embryology. So the unit what I am doing right now in human reproduction is embryology or developmental biology. What is embryology? It is the study of development of embryo from the time of fertilization till it is born. Right? Baby will be born, till that time whatever happens is embryology. Let us begin with the stages in embryology. Actually, in broadly we classify the development of embryo into different stages. First one we call it as cleavage. Second one we call it as blastulation. Third one we call it as gastrulation. Then we learn even under neurulation. After that, there will be the formation of 3 germinal epithelial layers. So these are the various stages we are learning. But in general, I am explaining with this. But in humans, how it happens, that I will be giving you in the heading cleavage. So these are the 3 sub-stages. Even neurulation happens after this. Now we talk about only cleavage. What is cleavage? Let's begin with this. Already you might have remembered that after immediately fertilization, division of the zygote starts by the formation of spindle fibers, which is because of the presence of proximal centriole, which is brought by the sperm, which divide into 2 daughter centrioles. Daughter centrioles produce the spindle fibers. They form the mitotic division. Begins at exactly immediately after the union of both male and female pronuclei. In the last class we learned it. It's called the amphimixis. Now, it is the next stage where the division of the zygote starts. It is called cleavage. How do we define cleavage? It is the repeated mitotic division of the zygote. Right? Means simple mitosis division happen, complete zygote is involved. So coming to the cleavage, I told you this cleavage is simple mitotic division I have here. But in this we have different types of cleavage in different organisms. The types of cleavage can come for 2 more questions. Right? You need to know what is cleavage or define cleavage, write the types of cleavages. First is cleavage is a repeated mitotic division. What are the types in this? Types of cleavage. First one is called as the cleavage involving the complete zygote. We call that as the holoblastic cleavage. See, remember the word holoblastic refers to the complete zygote is involved in the division. Right? Second one is meroblastic cleavage. So I hope you understood. Meroblastic refers to the division involving part of zygote or partial zygote. So only partial zygote is involved in division. That is called as meroblastic cleavage. This is the complete zygote is involved in the division. So two types of cleavages are there. One is holoblastic cleavage, another is the meroblastic cleavage. Then under holoblastic cleavage we have again two types. Holoblastic unequal cleavage and holoblastic equal cleavage. So what is holoblastic unequal? Means cleavage no doubt is the division of the zygote, and if it is holoblastic it is involving the complete zygote. Unequal refers to the type of holoblastic cleavage in which some areas divide faster than the other area. Therefore, one area divides and forms a large number of cells. Another area, few cells are formed because division is slower. Therefore, unequal cleavage, you can get a clue in this. Unequal cleavage is a characteristic feature of those organisms that contain yolk. If there is yolk present somewhere in the zygote, then that yolk will not allow the divisions to happen freely, because divisions happen freely if there is nothing hindrance, but yolk hinders the divisions. Therefore, in those organisms there is yolk present, there will be either unequal cleavage or it may be meroblastic cleavage. So let me explain how it happens. Right? If you give this as an example of frog that undergoes unequal holoblastic cleavage, what happens in this is, suppose this is the zygote, imagine, here is a nucleus present, okay, the lower portion is having a lot of, this is all yolk. So whenever divisions take place, in upper area divisions remain faster, fast, this divides, forming number of meroblasts, but lower divisions divides slowly. Lower area of the zygote divides very slowly. Therefore, this is called example for unequal cleavage, because in frogs, yolk is present at the lower end. And you must know one more thing, additional thing to this, let's suppose the neat portion. Types of eggs also. See, in this respect only I will give you. Types of eggs based on the amount of yolk. First one is amount of yolk. Amount of yolk, how do we classify it? So this is little depth of this, right? So if the yolk is present, it is normally, if the yolk is absent, it is alecythal egg. It starts with the presence of yolk, minimum quantity, moderate quantity, higher quantity. So based on that, we classify the eggs into three types. One is the first one, microlecithal egg. Microlecithal refers to minimum quantity of yolk is present, which is normally found in the insects. Little quantity of yolk is present. Second one is called as mesolecithal egg. Mesolecithal refers to the egg containing yolk which is moderate quantity, medium quantity of yolk is present. Example for this is your frog. And the third one is called as megalecithal or macrolecithal egg. This is the amount of yolk is too huge and lot of yolk is present. It is found in all the birds including hen. Okay? These are about the types of yolk, the eggs based on the amount of yolk they contain. But there is another classification, the type of eggs based on the distribution of yolk. See, I am talking with respect to this only, because to understand frogs egg you need to know others also. You can't just say okay frogs, you cannot by heart. Biology is not to by heart, to understand thoroughly, to know the subject. So distribution of yolk is a second category. First is amount of yolk, second one is about the distribution of yolk, how it is distributed. If it is distributed equally throughout the cytoplasm, means throughout it is present, it is called as isolecithal. Isolecithal, or we call it as homeolecithal. Means it is found equally throughout, it is present equal throughout the cytoplasm, right? So this is an isolecithal egg. Normally it is found in amphioxus. Second is, yolk is distributed only at the center. We call that as the centrolecithal egg. This is normally found in the insects. Central yolk is present. Then last word is telolecithal. See, the words are using. Telo refers to one end, right? Yolk is found at one side, one end, lower end it will be present. It is called telolecithal egg, which is, example is the frog, right? So now, based on this distribution of yolk, the divisions also take place, right? Why I told you this in regarding this, when I am talking about the types of cleavage, why did I explain you types of eggs? Is because the divisions will change according to the amount of yolk and the distribution of yolk. So, if I say unequal cleavage happens in the frog, that's because of egg yolk is found at the lower end. The divisions are slower than the upper area where there is no yolk. The divisions become faster. So therefore, in frogs, the type of cleavage is holoplastic and unequal cleavage. So cleavage we classify into first two. One is holoplastic, one is meroplastic. But under the holoplastic cleavage, we have unequal and equal cleavage. Now, where exactly equal cleavage happens? Equal holoplastic means complete divisions are equally throughout. It is divided the same way. It is called as a holoplastic equal cleavage. This happens in humans. I hope you got a logic in this. What is the logic behind this? In humans, eggs contain no yolk. If there is no yolk, divisions are equal all over. It's not that only one portion is dividing, because no yolk. No yolk means it is dividing equally. Therefore, in human cleavage, it is the holoplastic equal type. Right? So already I have told you these types, right? Eggs based on this, right? Microlecithal, mesolecithal, here nowhere humans are there. Why? Because humans contain no yolk. This egg is called alecithal. That means no yolk, right? Now, coming to meroplastic cleavage, I told you, meroplastic refers to the cleavage which is partial. Means some portions of the zygote is not involved in the division at all. Means divisions happen only at the area where is yolk-free container. Area containing no yolk, there will be division happening. So this happens in the insects. How? Just let me give you in broad way. Suppose in insects, if you observe, this is the egg. The yolk is present at the center. You can see here. I have written here centrolecithal egg. Example is insects. Right? In insects, you get the yolk at the center like this. Okay? So, if yolk is at the center, divisions are happening only at the periphery. The divisions are not happening at the center. Therefore, in insect it is meroplastic cleavage. That is, it is a partial zygote is involved in the division. Therefore, the cleavage is said to be the meroplastic cleavage in insects. So, now let me summarize. Cleavage, you know, it is a repeated mitotic division of the zygote. Next is, this cleavage is divisible into two types, major two categories. One is the holoplastic cleavage, second is the meroplastic cleavage. Holoplastic involves the complete zygote, meroplastic involves the part of the zygote in division. Now, coming to the holoplastic, again it is subdivided into two subtypes. Holoplastic unequal, holoplastic equal. What is holoplastic unequal? It is involving the full zygote, but some portions are dividing slowly and forming cells which are unequal sized. Right? Why? Because normally in such organism there is yolk present. The area containing yolk will hinder the division. Therefore, in that area divisions are slower compared to the yolk-free area. That is the example of frog. But in humans, the divisions of the cleavage is holoplastic equal. It is the example is humans. Why? Because zygote contains the cytoplasm only. There is no any kind of yolk present in humans. Therefore, all the areas divisions happen equally. So this Remember why it happens only in humans. Then coming to the meroblastic, I told you partial zygote is involved in the division, part of the zygote. Therefore, in these insects, divisions happen only at the periphery because yolk is restricted to the center, because the centrolecithal egg is having yolk at the center only. Therefore, this is the overall story of cleavage types, and types depend upon type of egg also. Therefore, the whole thing you just note it down, just pause, understand each of them, take a, if you want, screenshot and keep it as a notes. Some students today, they were asking me whether I am giving them notes. Listen, this is more than notes here. I am explaining everything live. If you want, I can give the written notes for your happiness. I will give you. But otherwise, everything is in detail. I am explaining. This will be always with you. Anytime you want, you just open it and observe and read, understand, draw and prepare by heart, whatever it is. By hearting, it should be less. But these names normally you will not get so easily. Little, but you need to revise these things. Okay. So this is about the cleavage outside. Let us move into the cleavage pattern. How exactly cleavage takes place? Right? General pattern I will give you with reference to frog. In humans I told you it is equal. Therefore 5 diagrams I will give you to show how exactly cleavage happens in frogs. Right? So let me just note it down. Let me start off with the cleavage pattern. How exactly it happens? What are the ways cleavage is taking place in all organisms in generality, and then particularly one organism, right? So in cleavage, one thing you should note it down, the divisions, if they happen from animal pole to vegetal pole longitudinally, that is called the meridional plane. So cleavage happens in two planes. One is in the meridional plane, referring to division happening from top to the bottom like this. It's a meridional plane, right? Then another one is, it may happen like this from the horizontal view. Then we call it as equatorial plane. This is the plane of division. It's the plane of division. Okay. So if it happens longitudinally or vertically, because the meridional plane happening, the divisions are happening in the meridional plane. If it happens in the center, in the horizontal plane, we call it the equatorial plane. Okay. So let me give you in briefing how exactly it happens. Up to 5 divisions I will give you. So first, cleavage normally in frog. Cleavage in frog, for the reference I am giving you. In all others, it may happen the same way or little bit different. It will be. So first cleavage, observe. See, at least you draw 5 such cells. Second cleavage. Okay. As much as possible, draw. This you can draw it with your compass, no problem, right? Then third cleavage. Right, all I will draw in one, say, let it be the same, there will not be increase in the size, therefore I am drawing almost equal. If you use compass, it will be easy for you. Fifth one is fifth cleavage, also same thing. Okay, you should know. If you can't see that, I will draw it again here. Okay, now, so coming to the cleavage, first cleavage, it happens longitudinally in the meridional plane from animal pole to vegetal pole. This one. Right? So this results in 2 cells. These are called the meroblasts. Right? First cell, second cell. Right? But whenever you are drawing, this will not look like a division. It's like a very plain diagram. So whenever you are drawing this, slightly give artistic touch, showing that it is showing the division. So it should look like division. Right? This is how it is divided. First cleavage is meridional plane. This is called the 2-cell stage. These are called as blastomeres. Now, sorry, meroblasts. These are called the meroblasts. Then second cleavage happens right angle to the first cleavage. First is happening, suppose this is a circle one, is dividing like this, is the first division. Second division happens exactly in the meridional plane, right angle to the first cleavage. Right? First is happening like this, second is happening like this. Right? Therefore, You can't see it. If at all I draw one more cleavage like this, you can't see. So I'm tilting and showing you the diagram. First cleavage is like this. This is the second cleavage. Can you make out? So this is how the second division is happening. This is the second cleavage. Okay. So learn it is for even NEET and all. This is very important for you. So this is the area where the cleavage groove is seen. So I am darkening this. Okay. So wherever there is joining, we just show a slight enlargement of this. So you can make out there are 4 cells formed. Very clearly 4 cells are formed. 1, 2, 3, 4. 4 blastocysts. It's called the 4 cell stage. Okay. This is a 4 cell stage. Then comes the third cleavage. Okay. So let me draw a little up so that the 5th cleavage also can observe easily. So let me draw up the third cleavage. Okay. Let me draw here third cleavage. So how is third cleavage happening? Again I am drawing here semicircular cells, and first 4 already cells are retained. This is the first division. This is the second division. Now what is third cleavage? These are all from the first only I have taken. The second cleavage is this. Third cleavage happens in the equatorial plane, dividing it transversely. But remember, I am explaining this in frog, and frog I told you the yolk is found in the vegetal pole, lower pole. Therefore, divisions happen slightly above the equator, not exactly at the center, slightly above the equator. So I will be drawing the diagram like this. Correct? So now here is the dissecting area. This I am going to shade it. So understand how to draw it nicely or easily. I am not just showing the ornamentation of this, but it should look real also. So wherever there is joining, I am going to just show slight elevations, right? So wherever I have just showed this, you remember how to show this. Wherever it has joined, I am just showing slight elevation, right? Here like this, again here elevation. So you can make out very clearly how are the cells. See, now the divisions are unequal, right? Where inequality of division starts in frog is in the third cleavage itself. So upper 4 cells, 1, 2, 3, 4, these are all called as micromeres. Important word is micromeres because they become small. They are smaller cells. Lower ones are large because there is yolk in it. All over here there will be yolk, right? This is the 4th, 5th, 6th, 7th, 8th. That is 4 cells above. This is the 5, 6, 7, 8 is the other side. This is the third cleavage, producing how many cells? 2 tiers of 4 cell each. That is totally 8 cell stage. Remember, 8 cell stage I am drawing here. Okay. Then comes the 4th cleavage I am drawing here. 4th cleavage. How it happens? 4th cleavage happens in the, see this is in the equatorial plane, but 4th cleavage happens in the double meridional plane. See the word I am using, meridional, you know it, cuts longitudinally. Double meridional means one division happens one way, then another division is followed by the same plane but right angle to the first plane, right? So first retain the original diagram, then draw remaining ones, okay? So this drawing will be carrying marks, and you should remember the drawing pattern also. So first 2 divisions I am retaining here. These are the first 2 cleavages. Then this is the third cleavage. What I told you, the next cleavage is happening in the double meridional plane. So first division is here. Second division is here. See, understand now. Can you make out? So here is the dissecting area here where all of them are joined here, right? Okay. Now here is, we are going to show the slight enlargement, right? Just a little bit time you waste on this because it's not a wastage, really it is going to be useful to you because it looks real biological diagram. Otherwise they are not just 2-dimensional pictures, they are all 3D pictures. Just you show some shading, it looks like a 3D picture, okay? You can make out here. Wherever there is dissection, there will be a small shade to be drawn here. like this, a shade has to be drawn showing that this area it is dissecting. See, you can very clearly observe, you can see as if like an apple is cut like this so many times. 1, 2, 3, 4, 5, 6, 7, 8. Okay? 9, 10, 11, 12, 13, 14. Then how many? 8 cells in each row, 2 rows. Okay? 16 cell stage it is. Right? So this is the 16 cell stage of cleavage. Understand, these drawings are very important. Just observe how there, and there is no increase. You should not increase the size of the zygote. Maybe while drawing, just approximately it may be looking bigger, but otherwise you are not going to increase the size. Then comes the last, 5th cleavage I am drawing here. Right? Just because I thought there will not be visibility, so I am showing it here. 5th cleavage happens in a double equatorial plane. See, there I said it is happening in double meridional plane, means 2 divisions are happening. Here it is happening in double equatorial plane. So what you are getting now? Original structure I will retain first. What is the original structure? You have the first structure. I hope it is seen, right? Then first cleavage, second cleavage, third cleavage, is the 4th cleavage. Then 2 equal equatorial divisions, 1 above the first division, 1 below the first division. See how many? 1, 2, 3, 4, 5, 6, 7, 8. 8 cells in each row, 4 rows are there. This is called the 32 cell stage. Okay, this is a 32 cell stage. Very important. This you remember. This is the total cleavage pattern. Understand, it is happening in humans it happens, but it happens in equal way. The pattern is same, but only thing is there is no inequality in the 3rd cleavage. It happens the same way. So till 5th cleavage it is actually traced. Afterwards it will not be traced. So this drawing you remember because it is like CET you think, NEET you think, all India medical exams, anywhere, even in agriculture, whichever part you are going in biological subjects, this is basics of that, right? Because embryology is a very important chapter. You are going to study how exactly the divisions are happening, and that's the reason I am just showing you with all this picturization. So understand, pause and check it how to draw them. Okay? But anyway, this drawing may be asked, or sometimes I'll ask you one each, which division? It is 8 cell stage. It is a 3rd cleavage. Which division from 32 cell stage? It is a 5th cleavage. Like that you have to learn each one, so become perfect in this. Okay? So just draw it and understand this. I hope you did it. Let us now begin the next one. What happens at the end of cleavage? So that's an important thing to remember, because this is the not only last revision. 5th cleavage is not the end of it. It will still divide continuously and forms bunch of cells, group of cells, and resulting in like one ball of cells, solid mass of cells, and very nice name given to this is morula. Right? Why the name morula is given? Because morula refers to the fruit of mulberry. This looks like a fruit of mulberry, so the name is morula. So how it looks like? So at the end of division, the cells look like they are all combined, right? Like all the cells I am drawing, central cells I am drawing a little bigger. These are all the cells which are formed at the end. They are all the meroblasts. And if you think of frog, they will be small at the tip, but in humans all cells are equal, right? Almost like the fruit of mulberry looks like this. Therefore it is named as morula. Morula is the fruit of mulberry. But in humans there will be one more layer around this. This is zona pellucida. Okay? This is about the nucleus of all the cells. This is the morula structure. Remember, morula is the solid ball of cells formed at the end of cleavage. This is morula. Okay? Now what happens? Next, the morula is going to convert into the blastocyst in humans. So blastula, I told you, it is a second step in reproduction, that is developmental biology or embryogenesis, in which the solid ball, I draw here, this solid ball of cells containing all these are blastomeres, and this is a zona pellucida. Let me label this. This is zona pellucida, and these are the blastomeres. Okay, this looks like a fruit of mulberry. Therefore, the name is morula. How to define morula? It is a solid ball of cells which are distributed equally without any cavity in the center. This is a morula. This actually resembles the fruit of mulberry. Why morula is? The mulberry biological name is Morus alba. Morus alba. That's why the word is morula, derived from that morus, morula. Right? So you should know. Simply, some words are not given. So most of the words have some meaning in normal language. So if it is only derived from Latin, you will not understand. Therefore, morula is a solid ball of cells containing blastomeres. Next is, this is the diagram. What is this? It is the kind of blastulation. What is blastulation? It is the enlargement of the complete morula with a central cavity called blastocyst. Right? Then cells go to the periphery. Right? In humans, we call this as a blastocyst, not blastula. It's called the blastocyst structure. What is the blastocyst consists of? The central blastocyst cavity is there. It's almost like a blastocyst cavity. But the two layers of cells are differentiated. You can see this is black colored one, what I have drawn is a trophoblast cell. Next one is called the inner cell mass. But trophoblast has another outer trophoblast. Here I have written as syncytial trophoblast. What is syncytial trophoblast? It is nothing but the trophoblast layer outermost containing no proper cell membranes and free nuclei, because the outermost layer of trophoblast, which is formed by the transverse division of this inner trophoblast, will lose the cell membranes and protoplasm and all will get distributed with the free nuclei. Nuclei will be freely located all over here. Therefore, the outermost layer is very sticky in nature. This is syncytial trophoblast. It has one important function. What is the function is? This helps in attachment of the blastocyst to the wall of the uterus during implantation. Understand properly. Right? Trophoblast, which is there inside, has proper cells. See, you can see here, this is one trophoblast cell, this is another trophoblast cell, this is another one. These have nucleus and cell membrane all. Therefore, we call this as the cytotrophoblast. Or trophoblast containing cells is cytotrophoblast. The outermost layer is called syncytial trophoblast. It has no proper cell membrane because they are broken down with the cytoplasm with the free nuclei. All this is xenocytic condition. Free nuclei are present outside. So this is about the trophoblast. Why I am talking so much about trophoblast? Because they are going to form in future the fetal membranes, fetal coverings, protective layers for the fetus. Therefore, trophoblast layer is very important for your one word question, 1 mark question. What are trophoblasts or what is the function of trophoblasts? Trophoblasts are the layers produced outside the embryo that in future form the extra embryonic membranes. Okay. Now, the major word I want to use is inner cell mass. You can see in the blastocyst cavity some cells are protruded. These cells are called inner cell mass. What is the beauty of them? These have the highest potential ability and can grow into a complete embryo. This is the future embryo, that is the inner cell mass. Okay, so this will differentiate, divide, differentiate and form the complete baby. Right? Now, one more important thing about inner cell mass I would like to give you now only. What is it? It is in modern technological, biological influences or new investigations have used this inner cell mass. What for? It is used as a stem cell. Then you will have one more word now. What is stem cell then? Stem cells are the highly potential cells that can grow into complete embryo. They have highest totipotency. Totipotency is potential ability of a cell to grow into complete organism. This inner cell mass has that ability. Therefore, in biotechnology, they use this inner cell mass to form the clones, new another organism, another human being. What they do is, in the stage when the mother is of this pregnancy, where her internal embryo is formed up to blastocyst stage, some of the inner cell mass are taken out and they can be kept as a stem cell preservation. You have to store the stem cells under preservation at liquid nitrogen temperature. This is the extra thing I am giving you. It won't come under this. I am telling you how it is useful right now in biotechnology. When you talk about biotechnology, again I will remind this. Okay. So these stem cells are kept in liquid nitrogen temperature, cryogenic preservation. Then in future, those cells can be used for the same one. If it becomes a young one and suppose it wants any organ, maybe a kidney failure happens, then those stem cells which are kept under preservation can be converted into kidney, can be converted into any other organ like heart membranes. The other parts where bone marrow, for everything they can be used as the first cells are called the stem cells. These stem cells are of highest research is going on in this. They are highest significant cells. They are more significant as the stem cells. Remember, that is the future. Nowadays cloning and all is banned, but otherwise cloning also can be done with these cells only, because these are the cells which form the future baby, future embryo. That's about the inner cell mass. Okay. Now, inner cell mass, what I told you, this is going to become a future embryo, and the trophoblasts are going to form the future embryonic extra membranes. Extra embryonic membranes are the fetal membranes. Now, this diagram in board, you can get it for 2 mark, just to draw a label, or 3 mark, draw a label and explain the parts of blastocyst. Now, the nice thing about this is, this can be, on this you can build the CET and NEET portions. What is that? What is syncytial trophoblasts? How are they formed? What is the significance of syncytial trophoblasts? Or what are cytotrophoblasts? What are the inner cell mass? What are the stem cells? So like that, everything can be understood based on the basic subject, that is blastocyst. So now you pause this and draw the diagram. This drawing is very important for you. Easy also. No much, lot of details you don't have to draw. Just draw one diagram, some inner cell mass and trophoblast, labeling with the blastocyst cavity will give you 2 mark or 3 mark question. Okay, just draw this now. Okay students, I hope you have drawn it. Now, I told you some of the questions. This is almost end of major portions. Some of the questions I will discuss before I enter into the details of this. So what are the questions you can expect till now, what all we have done? First thing, from the reproductive structures. Male and female reproductive structures, 5 mark question. Mammary gland, 5 mark question. Right? So let me give you list the questions what are expected in the board examination. I am not talking about need portions. I told in between only. Explanation I did while explaining the major subject. Okay. So first thing, what are the this you remember when we are going to give the test. These questions I will give you. Not only me, any board examination you go through, these questions are there. So what are the expected questions for the board? I am giving you those questions. First thing about the male and female reproductive tract. Female reproductive systems. How much? 5 mark question. It's almost like pakka question. It will come. Right? Second is about the structure of the mammary gland. You could have seen last year they have given this question for 3 marks. Before that we given it for 5 marks. Male and female reproductive system, 5 mark question. Third one, Apart from this, what else I told you? Section of the testis. Testis TS is a 5-mark question from the practical part. Right? Then, fourth question. Spermatogenesis 5-mark question and oogenesis 5-mark question. Oogenesis. Okay, 5-mark question. Okay. Next, fifth one is structure of the sperm or structure of the egg. Egg will carry 2 marks. Structure of the sperm with explanation 5 mark. Otherwise, it's a 2-mark question. Draw a label. Right? Then sixth one. Then we talk about the Graafian follicle structure. So, Graafian follicle diagram from the practical is rated for 5-mark question. Right? Then seventh, with all which we have done, see so much we have completed already. Then we are talking about the process of fertilization in humans. Fertilization is again a 5-mark question. If you really want to fetch marks, draw them. In between I drew around 4 or 5 diagrams for penetration, acrosomal reaction, everything you draw. 1, 2, 3 diagrams, amphimixis. Then it will give you marks 5-mark question. Now in embryology, first one is cleavage. Cleavage and types and patterns. It may be 2-mark or 3-mark or 5-mark question. Based on how big is the question. What are they expecting? Is it only cleavage definition? 1 mark. Types of cleavage 2 or 3 mark. With the pattern of cleavage, again 3 mark. But total cleavage, all the diagrams till 5th cleavage is a 5-mark question. Then next is the structure of blastocyst and structure of morula. It's a 2 or 3-mark question. So these many things we have covered. Learn this, and whichever test you are facing, this becomes so simple for you because almost you know the subject also, you know the questions also, and you can very nice advantage you can see in children. You can just pause, draw it, pause, read again, pause and listen again. So easy, right? So actually technology has improved a lot and you can do all new things in this, right? Now let's now move on to the next unit. Okay, but first you just note down the questions. Continue with the germinal epithelial tissues. Why the germinal layers are talking about? Once blastulation ends, the embryo enters the gastrulation stage. Gastrulation is development of another cavity called archenteron by reducing the blastocyst. That's called gastrulation. Means there is a blastula cavity, blastocyst. A new cavity develops with one blastopore opening, and then it's like almost this way. Suppose this is the blastula, right? This is the blastocyst. This is the blastula structure. Now a small intake of cells, the cells get tucked in and the cells start moving into this. This is called the blastopore cavity. The blastopore develops and the cell starts moving inwards. Cells start moving inwards and this develops one more cavity. We call it as a gastrocele or archenteron. This in future going to become the gut cavity of the gastric gland or gastric tissue or the gastric gland. Not gastric gland, it is the alimentary canal. Gut is the alimentary canal. This is going to become the canal of the gut or gut cavity. That is the gastrocele. Blastocele will reduce later. So after the end of gastrulation, this we don't have it much in detail about gastrulation, we have to learn. At the end, 3 different primary germ layers develop. Primary germ layers are those layers that are going to form various parts in the future. Right? Even these can be seen at the blastula stage itself. It is called the fetal blastula. If you stain the blastula with various stains, then different colors will be taken up by different cells. Different layers are seen. But at the end of gastrula, the 3 layers are distinguished, namely ectoderm, mesoderm and endoderm. Ectoderm is outermost layer, mesoderm is the middle one, endoderm is inner one. The most important thing is they will ask you the questions on what are their derivatives. What are the derivatives of various primary germ layers? That I have listed here. Just go through them. You will have to remember pakka what is this going to become in future. Like ectodermal derivatives. Ectoderm is outermost layer. No? It forms epidermis of the skin. It means outermost layer of the skin. It is epidermis of the skin. And also epidermal derivatives. Even horns, hooves, nails, hair, everything develops from the ectoderm. Right? Ectoderm from the epidermis of the skin, hairs, horns, feathers of the birds, and hooves in animals. All of them they are formed in the ectoderm. Right? Apart from the nervous system, the entire nervous system, brain and the spinal cord, both are found by the ectoderm itself. You have to be very, very particular because these are the separate questions, 1 mark question they are asking. They are going to ask you, like asking, what is the derivative of ectoderm? What are the derivatives of ectoderm? You write around 3, 4. Or else they may ask you, brain is derived from which primary germ layer? Then you have to write, it is the ectoderm. Then, in eye, retina, lens, the cornea and conjunctiva. That is retina, where the focus is formed. Lens, the first glass thing in the front. And cornea, the side one. Conjunctiva, the white part. The whole thing of this eye is formed by mainly the ectodermal. Ectodermal is eye, complete almost eye. Then comes the salivary glands. You know already salivary glands, first piece we have studied. The parotid glands found at the below the ear. Then submaxillary glands below the lower molar teeth, the premolar teeth. Then the some more sublingual glands present below the tongue. All the salivary glands are formed by the ectoderm. Ectoderm derivatives are all the types of salivary glands. Then enamel of teeth. So you must be knowing again first PUC teeth structure you have learned. Outermost layer of the one which you get the color for the teeth is because of the enamel. Right? Enamel is derived from the ectoderm because sometimes you will see in genetic problem enamel will change. Some will have light whitish color, some will have light yellowish color. This is enamel. It is not of the total teeth. Teeth internally everybody will have same. But enamel is derived from the ectoderm. Right? Then pituitary gland. The pituitary gland that present in the brain at the hypothalamus area that actually controls all the endocrine glands is pituitary gland. It is also derived from the ectoderm. Then pineal body. Pineal body is the roof of thalamus producing one protrusion called pineal body, pineal gland that is going to control your sleep-wake cycle and the pigmentation. That is pineal body. So you remember all the derivatives of ectoderm. Let's move on to mesoderm. Mesoderm is a middle layer. This is going to form almost all the connective tissues. Like what are those? Dermis of the skin. Just below the epidermis will be dermis of the skin. Muscular tissue. The complete muscular tissue like striated muscles, it may be the smooth muscles, it may be cardiac muscle, everything is formed by the mesoderm only because it's a almost connective tissue. Then connective tissue, complete areolar connective tissue, cartilage, everything is formed by the mesoderm. Then bone, complete endoskeletal system, either it is a bone or cartilage. You can add up here cartilage also. Some will have endoskeleton made up of cartilage. Cartilage and bone both are made up, produced by mesoderm. Then heart and the blood vessel. The complete heart membranes and the blood vessels completely formed of the mesoderm. Remember, because so many times you will not suddenly get it. So you at least try to by heart or remember how they might have derived from. Mesoderm derivatives are heart and blood vessels also. Then kidney, the complete kidney, the two kidneys located at the vertebral, near the vertebral. So we are talking about the germ layers, right? Primary germ layers. The fate of various germ layers I am talking about. We already explained about the ectoderm. Let's continue with the mesoderm. I told you that dermis is the just layer beneath the epidermis of the skin. Muscular tissue, as already I discussed, mesoderm produces all the connective tissues, right? Muscular tissue is also connective tissue. Then comes connective tissue. Connective tissue have complete areolar connective tissue, right? And all the connective tissues will connect part to part, right? Like areolar connective tissue. Then bone and cartilage both form the endoskeleton. In higher animals, mammals and all, the bone is forming endoskeleton. In lower ones like fishes and all, cartilaginous species, cartilage forms the endoskeleton. Then heart and blood vessels, complete heart and blood vessels are made up of, are produced by mesoderm. Kidney the complete kidney. Then urinary and genital tracts. Means urinary tract is ureter, genital tract is fallopian tube, and some are vas deferens. All of them are the genital ducts. All of them they are formed by the mesoderm. Then come to the last one, endoderm. Endoderm on the complete gut. That is a digestive system, complete from mouth to esophagus, the stomach and the small intestine, large intestine, rectum and anus, everything. The complete gut is nothing but the alimentary canal is formed by the endoderm. Then glands of the stomach and intestine. You know the stomach and intestine glands. The stomach glands are gastric glands. Intestinal glands are Brenner's glands. Brenner's glands are crypts of Lieberkühn. Hope you remember crypts of Lieberkühn, and the stomach glands are gastric glands. Okay. Then tongue. The complete tongue is made up of produced by endoderm. Then lung, trachea and bronchi. So lungs, the complete respiratory system. Its complete lungs are made up of this. Trachea is the complete voice. What is called the sound box. That is the sound box is larynx. Then comes trachea. It is the air passage. Then bronchi is a branched trachea. Then urinary bladders. The urinary bladder, single bladder where the urine is collected. And then gills in the fishes which help in breathing. Liver and pancreas, they are also the digestive glands. They are made up of, they are produced from endoderm. Then thyroid, parathyroid glands lining of the vagina and urethra. These you are supposed to understand thoroughly. These are 1 mark questions, 2 mark questions or 5 mark questions. Suppose they ask you liver and pancreas are formed from which germ layer, then you must write endoderm. Like that everywhere you will be expecting a question. You need to by heart all of these. Don't mix the things. Most of the times get mixed up. At least by default you should know ectoderm from the outer layers and nervous system, mesoderm from the connective tissue, endoderm from the internal duct, like digestive duct, complete digestive system. Then along with the liver and pancreas. This is in general you remember, but in specifically everything you have to by heart. You better read it by heart. Okay. So this is about the various fate of various germ layers. Okay. Understand this? Note it down or take a screenshot. Keep it. Let's continue with the next process. Once we have learnt about primary germ layers, we are now talking about the next, how exactly embryo is fixed. Actually, we call the first process, once the blastocyst is formed, then it will move through the fallopian tube into the uterus. Uterine wall will become thick by the help of the hormone progesterone produced by the corpus callosum. Already I discussed with this. Along with that, it will go on increasing in size with more and more blood vessels. Then from a thick mass or blood vessels bed is formed. In that, the fertilized embryo will go and fix. That process we call it as implantation. Very important word is implantation. So implantation is a fixation of the blastocyst to the wall of uterus. Right? In short, I'll just give you the diagram. Suppose this is the uterus. Okay. Already you know how it will be. Okay. And this is the endometrial tissue. This is the cervix. See, again and again I will be drawing just to show that how easily you can draw this. Okay. This is the endometrial tissue. This is the cervix, cervical canal. Okay. And here comes the vaginal area. And here is the upper endometrium. Okay. So this blastocyst comes from this fallopian tube because fertilization has happened in this portion. What is this portion? This is the mid of fallopian tube, fertilization happens and blastocyst is formed. This will be rolling and it comes and fixes here. Okay? The fixation happens somewhere in any part of the wall, either in the top or the sides. Imagine it is fixing at this side. This is a blastocyst I am drawing. It has one important property. I told you it will be very sticky because of the outermost layer which is made up of syncytial trophoblast. Therefore, it will be sticky and it will stick to wall of the endometrium. Now, understand one very beautiful thing. This blastocyst or external layer, that is syncytial trophoblast, they release a kind of lytic enzymes that will dissolve the wall of endometrium, and due to which the endometrium, it forms a nice pit here, and this whole blastocyst will enter into this area and get inside this. It is now our blastocyst. Now, because of the progesterone hormone, lot of new tissues are developed all over here, and almost at the 7th or 8th day of the fertilization, this is fixed. Implantation. This process called implantation. Now, because of the hormone, I told you, hormone name is progesterone. Progesterone secrete or helps in the formation of thick new layer of endometrium all over. This is a new layer formed, right? And this newly formed endometrium is called as decidua. Decidua. And this is our blastocyst. It has implanted. This is a blastocyst. Then this is all newly formed endometrium. We call that as decidua. Some of the decidua that is covering the complete of blastocyst, we call it decidua capsularis. The one which is found at the base of this is called decidua basalis. And all over other areas also endometrium is formed. This is going to degenerate once the fixation happens at one side, other side whatever is newly forming, that is going to disintegrate, degenerate. This is called decidua vera. So what all you have learnt in this? Implantation is the sticking or fixing of the blastocyst to the wall of the uterus. How it will happen? It will first stick to any part of the endometrial tissue, which creates a small pit by producing certain lytic enzymes and get inside the pit. And at that time, that is 7 to 8 or 8 to 10 days of pregnancy or fertilization, the embryo is not at all seen inside the uterus, it will be inside, within the decidua. So new endometrium is formed. This is called as decidua. This decidua covers the blastocyst, and from the base of the blastocyst and all over the uterus also there will be formation of decidua. Accordingly we name them. Decidua capsularis, outermost layer, decidua basalis present at the base. Decidua vera is the other type of decidua or the endometrial tissue that is going to disappear in later stages. So this is about implantation. Just understand how implantation takes place, because now we are going to talk about the next portion, that is how is the embryo and placenta developing. This is a very important thing. After this embryo begins to grow, develop, right, and a placenta is formed. So let me draw that and understand how beautifully the fetus will be located within the mother's womb. Okay. Let me start with the structure of placenta and the embryo. Embryo and the placenta I am going to draw now. Okay. So first, let me talk about the overall endometrial tissue. Imagine this side it is fixing. Okay. Only that portion I will enlarge. Later I will show other areas. So this is the original area, what we call it as perimetrium. This is the perimetrium of the mother's womb. Then comes the myometrium, another layer. This is the myometrium. Above that is the endometrium, right? This is the endometrium. This is like this. Imagine it is fixing over here. Okay. What happens for fixation? The embryo, whichever is formed, that embryo will have the outermost various coverings are developed. Then what are the coverings? So let me draw. This is the extra developed endometrium. Now the new layers, the layers of the fetus are developed. This is the outermost layer of the fetus or extra embryonic membrane. We call it as a chorion. This is called the chorionic layer. This is chorion. Okay. Chorion will enter inside this endometrial tissue and form number of such finger-like projections. I hope you can understand how this is drawing the nutrients from the maternal tissue. This is our villi of chorion, they are called the chorionic villi. These are chorionic villi, you can see here. So I am mixing both of them. You can see this is the endometrium of the mother, or we call the decidua, and this is our fetal membrane. First fetal membrane has entered into it, become a part of the placenta. These are called chorionic villi. This villi is involved in absorption of nutrients. Chorionic villi. Okay. Now, very important thing is the chorionic villi are not directly penetrating or breaking the blood vessels. What happens is whatever mother is having blood vessels, right, supply will be there. These blood vessels are broken down by the chorionic villi enzymes, due to which this area is full of blood. Maternal blood will be there. All over here it is red blood found here. Directly chorionic villi come in contact with the blood, but the fetal blood will not combine with this. Okay. So this is the chorionic villi and this is the maternal blood. Okay. In this they are dipped. This is the maternal blood in which chorionic villi are dipped and draw the nutrients from that. How do they do that? In the chorionic villi, number of blood vessels will be present. The diffused nutrients from the maternal side or from mother's side, that will be transported via these blood vessels. These blood vessels of the fetus together they join and form a small thread that will attach to the fetal body. This we call it as the umbilical cord. The umbilical cord is nothing but the one which is attached to the navel region of the fetus. And here is our fetus. Fetus is, okay, this is the baby. Okay, it is attached to its navel. Navel is the one in the tummy area. Okay, this is the fetal face, and here are its fingers, small ones. Here is its hind limb, and here is its tail, right? And there is a big eye orbit will be there, right? This is the structure. So understand, this is the complete structure I have drawn about the fetus. And here, let me give you the details of the other parts. Here is the uterine portion, right? This is the uterine portion. So let us discuss then how exactly destroying nutrients, what is placenta. Now you can see that this is the outermost chorionic layer. This chorionic layer is forming the number of finger-like projections that enter into the maternal blood and draw the nutrients. Whatever is diffused and by diffusion of food is entering, that will be transported through these blood vessels. The blood vessels through which food is transported from the maternal blood to the fetus. Fetal blood vessels I am drawing here. These are the blood vessels of the fetus. Okay. These blood vessels of the fetus are called as allantois. Allantois is another type of fetal membrane, that is extra embryonic membrane. First extra embryonic membrane is chorion. Second one is allantois. Then next is about one more layer is there just immediately outside the fetus, like this. This area or this layer, whatever I have drawn here, this is called as the amnion. This is the amnion. This will contain amniotic fluid in which the fetus will be moving around. It will be fetus. The only area where the fetus can get right pH, right nutrients, everything. It will be safe inside the mother's womb. Because of this fluid, we call it as amniotic fluid. Amniotic fluid, which is present in the amnion. Amnion is a sac-like structure. So, what are the membranes I told you? One is chorion, one is allantois, one is amnion. Apart from that, there will be another one. This is called as the yolk sac. This is the yolk sac. This is also another fetal membrane, extra embryonic membrane. So, there are 4 extra embryonic membranes. One is chorion, second one is allantois, third one is the yolk sac, Fourth one is amnion. 4 membranes are there formed by the, remember, trophoblast cells of the blastocyst. Then let's talk about the placenta. So this whole thing from here till here, it is a placenta. This whole thing we can consider it as a placenta. Right? What are the contents of placenta? This is the maternal tissue and maternal blood present. Right? Therefore, this is the maternal part of the placenta. So maternal part is the endometrium tissue is there and maternal blood is there. That forms the major part of the placenta. But apart from that, remember, apart from that, fetal tissue also from the placenta. See, chorionic villi are entering into the maternal tissue or endometrium. They are penetrating, and the food and other nutrients brought by the maternal blood are dissolving or they are diffusing across the chorionic villi. And these nutrients are taken up by these blood vessels. You can see here in every chorionic villi I showed some blood vessels, right? These blood vessels are taking the nutrients that are absorbed. There is no blood to blood contact. Remember, that's why there is no blood group problem. Although mother is A blood group, baby is B blood group, it can grow. There is no antigen antibody reaction happening because there is no blood to blood contact. Remember this properly, because most of the students at the end they will ask, how come my mother is having A blood group and baby is having B blood group? There may be egg agglutination, it will not happen. Because you can see where exactly is the barrier. There are only chorionic villi, they are separate, they are only drawing the nutrients. Diffusion takes place. Some of the antibodies may transport, but nutrients, respiratory gases and all will transport through this. Therefore, there is no blood to blood contact. So, the whole thing from here till here, we can call this as the placenta. So, placenta is this whole thing from here till here, it is placenta. Right? So, placenta, how do you define? It is an organic connection between the fetus and the mother, formed by both maternal tissues and the fetal tissues that help in supporting and the growing of the fetus or the embryo. That is the definition of placenta. Now, so this is placenta, the whole thing. Maternal tissue also is there, fetal tissue also. This is placenta, right? Now, how do you describe placenta? Very important question is, how do you describe the placenta? Very important question. So, human placenta is described with 5 terminologies. What are those terms? First term, human placenta is said to be chorionic placenta. Why? Because chorionic villi form the part of placenta, you can see here. That's why here chorionic villi are there, it becomes part of the placenta. So the human placenta is described as a chorionic placenta because placenta is formed by chorionic villi or the chorionic membrane of the fetus. So it's called chorionic placenta. What is the second term? We call the placenta as allantoic placenta. What is allantoic placenta? The placenta is also made up of allantoic blood vessels. See here, all the blood vessels are allantoic blood vessels that form the part of the placenta. Therefore, it is called the allantoic placenta. Then third one, it is called as metadiscoidal placenta. Why? It forms a small disc, half disc attached to one side. Meta-disc-like structure, half disc-like structure attached to one side. Not everywhere, only one side it is attached. So it is called the metadiscoidal placenta. Then the fourth point is, it is called as deciduate placenta, very important word. What is deciduate? Decidua is nothing but a maternal endometrium tissue which is newly formed. This becomes a part of placenta, and how do you say it's a placenta? Because whenever the childbirth happens, that time this newly formed decidua also will be scraped out. The whole thing will come out during the childbirth. Therefore, we call this as a deciduate placenta also. Then the fifth word is hemochorial placenta. See the word hemochorial. See, these words you should never forget. Hemochorial refers to, hemo is blood, chorial is chorionic villi. That's actually histological explanation of placenta. Number of chorionic villi are dipped in the maternal blood and draw the nutrients. Therefore, the placenta is called hemochorial placenta. Correct? So 5 terms you should never forget and understand their meaning. First, chorionic placenta. It is a chorion, the fetal extra embryonic membrane formed a part of placenta by forming chorionic villi. Second, allantoic placenta, because allantoids are the blood vessels of the fetus or fetal membranes that will also become part of the placenta, therefore it is called the allantoic placenta. It is a structural explanation. Third one, it is called metadiscoidal. It is showing the attachment and shape. Half disc-like thing present at one side. It is called a metadiscoidal placenta. Then comes deciduate placenta. Decidua is a maternal endometrium tissue. Also is a major part of the placenta which will be removed during the childbirth. It is also called a deciduate placenta. And last but not the least, it is hemocorial placenta. Hemo is blood, corial is chorionic villi. So chorionic villi dipped in the maternal blood and draw the nutrients, so it is called the hemocorial placenta. Then, so understood now? Two things. One is extra fetal membranes, 4 of them, chorion, amnion, allantoids, yolk sac. Yolk sac is non-functional, it becomes it is a vestigial organ in humans, but it is there. So these 4 of them are the extra embryonic membranes of the fetus. Next is about the placenta. What is the definition of placenta? I told you. Now, what is the way you describe placenta is by these 5 things. Now, last one is the functions of placenta. This is a very important question. Board also they will ask, and any other competitive exams also they will ask. Now, before that you draw this diagram and pause it, understand what it is, then repeat the drawing, then only I will give you the explanation about the placental functions. It's a 5-mark question. Okay. First this you draw and understand how it is, then let's go to the functions of placenta. So once we understand the structure of placenta, we need to know now what are the important functions of placenta. Okay. So as you already know, functions of placenta mainly is for the helping of growth and development of the fetus. Now, what all it will do? First thing, placenta will nourish the growing embryo by supplying the nutrients whatever mother has taken in during her food intake. After her digestion, the food is converted to glucose, fructose, or amino acids, or fatty acids. Everything will be in a simplified form. That blood will be supplied to the fetus through placenta. So placenta acts as a food reservoir which is transporting nutrients in simple forms. Second is, it helps like a lung. Placenta is like a lungs. How? It will transport the oxygen from the maternal blood to the fetus and remove the carbon dioxide which is produced by the placenta from the fetus to the placenta. So placenta helps like a lung by transporting oxygen to the fetus and removing the carbon dioxide from the fetus. So it acts like a lung. Third thing, it actually helps like a liver. You know how? Normally placenta will store sometimes the glucose into glycogen. It converts glucose into glycogen and stores it. Okay. Whenever, means if the mother is overeating and all, extra whatever glucose is there, it converts into glucose, glycogen and stores in the placenta. Whenever the fetus is in requirement of the nutrition, then convert the glycogen to glucose and provides it to the fetus. So beautiful. So it's a nature. So it's like a liver. Placenta is acting like a liver. You remember the words and write the explanation. Then placenta is like kidney also. It removes the nitrogen waste found in the fetus through the maternal blood. It will transport it out. Then apart from that, it will also help in producing certain hormones, because of placental hormones. What are those? One is the hormone called human chorionic gonadotropin hormone, HCG. HCG is called human chorionic gonadotropin hormone produced with the placenta. So these are the placental hormones I am writing. Placental hormones. One is human chorionic gonadotropin hormone. Another one is CGP. CGP is chorionic growth prolactin. Right? These are the important two hormones produced by the placenta for the development of the fetus. Right? Now, so it acts like a hormone producing endocrine gland also. So I told you placenta acts like a digestive, like a food storage organ by transporting the nutrients. It acts like a liver. It acts like the lungs. It also acts like endocrine gland. Right? And it helps in the development of the fetus, and it will give the certain, like whole fetus can be rooted, deep rooted, and it will be fixed properly. So that's how placenta helps. But there is one negative, some of the negative things are there in the placenta. One is it cannot distinguish between the pathogens or any other protein. Therefore, most of the virus like mumps, measles, HIV and all will be transported from the placenta to the fetus, and fetus will be highly affected. You must be knowing, mother having HIV, she will pass it to the fetus because fetus will get it through the placenta. Placenta don't act as a barrier for the viral transportation. And even mumps and measles, if the mother is affected, fetus will get it, and the young born baby will be having problem with the pancreas. They will have pancreatic problems, or sometimes they will be born blind because the virus affects the fetus very badly. Therefore, it's a negative point about the placenta. One more important negative part of the placenta is the alcohol or drugs, whatever, if the mother is taking in, they will be easily transported to the fetus. That is resulting in the fetal nervous system problem. Right? Fetus get Alzheimer's or some other problems when mother is alcoholic or mother is a drug addict. So fetus will be badly affected if mother is a drug addict. One more thing. Have you heard about erythroblastocyst fetalis? This is a disorder in which Rh incompatibility is involved. If mother is Rh negative, fetus is Rh positive, and if she has already, she is Rh negative, she don't have Rh factor at all. During second pregnancy, if she has first pregnancy with Rh positive fetus, then she will have a lot of positive antigens in her blood only. So when she will carry the second fetus or second time she is pregnant, that time all these Rh positive antibodies produced against the Rh antigens will be transported from the placenta to the fetus, causing the erythroblastocyst fetalis of the fetus. That is, erythroblastocyst means erythroblasts, that is RBC cells of the fetus are broken down. There will be agglutination, clumping, and fetus may get dead. It get killed. That is erythroblastocyst fetalis. Very important to understand where exactly the problem appears. If she is Rh negative, she gets Rh positive fetus only is a problem, right? And normally Rh positive fetus is formed when her husband is Rh positive and she is Rh negative. Positive factor is dominant over negative. Therefore, the fetus will get positivity. Therefore, Rh negative mother, A negative, B negative, O negative, AB negative, any negative blood group, if she is there, if she gets Rh positive fetus or A positive, B positive, AB positive, whatever it is, then there is a chance that second baby will not survive because of the erythroblastocyst fetalis. Very important thing you have to know, because this is the one which is affecting the future of the baby. Right? That's a death also. The baby will be having stillbirth. That's a negative factor or negative effect of placenta. So that's on the placenta. You will get this in a 5 marks. Now last thing I will describe, and one more thing you must know. One more thing is amniocentesis. Very important word I am giving you. Amniocentesis. That is, this is one kind of a test. It is a test in the pregnant woman. Doctors will take some amount of amniotic fluid from her amniotic sac when she is pregnant, and it is used in checking the fetal chromosome number or health of the fetus. But amniocentesis is actually good to understand whether the baby is normal or it has any genetic disorders. But nowadays the people are misusing it for identification of sex of the fetus, due to which most of the time the fetal, normally the doctors, I don't know whether doctors are involved or not, but the parents are insisting doctors to abort the female baby. That is, female feticide is happening mainly because of somehow they will come to know the baby is the girl baby. Then most of the parents don't want to have girl baby, which is a wrong opinion. In the modern era, this kind of thing should not happen. But anyway, amniocentesis is one of the way by which you can identify the sex of the baby also. But anyway, this is causing some more problems. If the parents only are educated, they don't differentiate when the boy and the girl, then this will never happen, right? But still, the female feticide is more increasing, or it's happening in most of the villages where there are not educated people. They don't have idea about why not to allow the girl baby to grow or let it survive. That's a problem. Anyway, by learning biology, you will come to know what's going on in society. How can you help the people who are downtrodden, who don't know anything? At least you can give them some knowledge about this. Okay. So keeping that aside, last thing I'll explain you is about the development of the fetus. What are the stages in which how it is growing, how it is developing. Around 4 weeks or 5th week, almost I told you, the implantation will happen, or 6 to 7 days implantation happens. Then how it is developing. In the first month of development, large head will be seen. Always head will be very big. When the fetus is growing, head becomes bigger, and limb buds are growing. Small baby like buds will be growing. Then heart beating starts in the first month itself. Heart beating means the parents, the doctors can identify by using stethoscope the fetal heartbeats also. By putting the stethoscope on the womb of the mother, they can identify whether the heartbeats are heard. They will be heard in the first month. And nervous system also develops in the fetus. At that time only they start reacting to the environmental stimuli. But it will have a small tail, right? Which is in the first month, which almost all the embryos of the mammals and others are same. They will have small tail. Then second month of the pregnancy, the bud-like limbs, they will develop small fingers. Suppose this is the bud-like limbs are there, then these limbs will develop small fingers like this, right? The fingers develop, and toes and the feet also develop. Skeleton begins to grow, means thin skeleton is seen if you scan the baby, right? And cartilaginous skeleton, and mainly it is cartilaginous, not the bony skeleton. But eyes like things are formed, the big openings are formed, they are closed. They don't have any eyelashes or eyelids, nothing. Okay? A closed eye-like structure is formed. Then third month of pregnancy, fetal movements begin. Mother will not feel on the third month only, but fetus start movement within the womb, right? Then organ development begins, means like small liver and other parts start growing. Then fourth month of pregnancy, head will be still large, big only. Facial features develop, small nose like thing and ear like thing and all this start developing. Lips also will be formed. But the baby's skin will be transparent, translucent, it will not have that color how the baby will have it. It will be like a transparent plastic like color, right? Then skin will be translucent, gender will be distinguished. The fourth month only you can identify genital organs will develop. The boy baby will have developed the small testes and all. The girl baby will show the pubic organs and all, right? Then fifth month, very nice thing is, at 5th month, baby will be fully covered with fine hairs, brown hairs, all over the body, like hairs, lot of things, hairs will develop like, teddy bear. Like, they are called the lanigo. Lanigo is the name of those fine hairs developed all over the body. Their whole body will be filled with those hairs like a bear, teddy bear like thing. Then, fetus looks like a tiny human being. Now the baby develops the human being features, like a head and small neck and the body and limbs and all this start developing. Still tail will be there. It start reducing time to time. Then the 6th month, and even fetal movements are felt by the mother. Mother start feeling that, oh, baby is kicking, baby is moving this side, that side. Mother will start feeling only in the 5th month. Although it starts movement in the 3rd month, baby's movement are felt by mother very easily, and they begin now, right? Then eyelids are formed. Eyes, I told you, it will be just like an opening. Now the lids are formed. Small lids are formed, but the eyes are closed. Then 6th month, lungs and digestive system, which are not fully developed, they are still not fully formed, but they are getting formation. Then hairs are still present. That is called as the lanigo. I told you lanigo is a fine hairs present all over the body. They are still present. 8th month, baby's head moves downwards. Means baby will be earlier than I drew the diagram. It will be facing up. At 8th month, it will move down. Okay, it will be almost attached to down part in the cervical area. It will just make the head down, right? That's the first thing, downward position. And eyes, lids and all formed, then open. Eye will be open. On 8th month, if you scan or observe the baby, his eyes are open. Then 9th month, body hairs are disappearing. No body hairs left. Then fat deposition starts in the body. Then it is ready to get delivered, because the complete uterus will be filled with the baby. No space will be there for its movement also. Therefore, in the 9th month, because the period of pregnancy, we call that pregnancy period is of 9 months, few days, around 10 days, plus or minus 10 days we take. That is the period of gestation. At this time, it will be ready to get born. Right? So, apart from that, what you have to know is, what is gestation? What is gestation? It is the period of pregnancy. The time of fertilization till childbirth is called gestation. In a 9 month plus or minus plus 10 days, the baby will be born. By putting the head will be down and open up the cervix through the birth canal, it will be released. At the time of childbirth, the hormone oxytocin is released by the mother's pituitary gland. Oxytocin is the hormone called as a quick birth hormone. Remember, so many things I am saying. Oxytocin is a hormone produced at the time of childbirth, which will help once it's produced, rhythmic contraction of the uterus happen and it push the baby down and helping in easy childbirth. During childbirth, what happens? The amniotic membrane breaks, amniotic fluid comes out, along with that the baby's head will come first, because the biggest organ is head of the baby. Along with that, the whole baby will come out, along with the placenta is also moving out. So after that, umbilical cord will be cut and the baby will be made independent. Then that's the end of the childbirth, and baby will be first allowed to breathe easily. Then after it start crying, its survival rate is 100%. Right? That's how childbirth takes place. That's how we all are born. So beautiful is the subject to understand, right? Because we don't know how we are born also. Now we have understood all this. Writing the examination is like a second part of it. So learn it thoroughly. It's very interesting. Understand all these things and refer any other books also. Whatever you have, you refer any other book and understand, or any of the such videos also you watch. Childbirth videos or development of the fetus. They do fast forward and show what all the things are happening. It's a beautiful thing. It's like a human female body is like a magical thing. Right? It's a nature. Real, we call it a motherhood. It's a precious thing. She will have one kind of hormone called LTH, luteinotrophic hormone. During pregnancy, she can feel the baby's movement and she starts loving the baby. This is why we call it a motherhood hormone. Right? That's why she will be sentimentally attached to the fetus or her own baby when she is pregnant. And after the childbirth, immediately oxytocin and LH hormone, or both of them, that is a lactating hormone, are released. Mother's milk glands or mammary glands start releasing the milk. The baby start feeding the milk, and baby survival rates are improving. Because of that only, mother will remain healthy and fetus will develop all disease. Actually, even the placenta will provide a lot of antibodies when the mother is pregnant to make the baby strong against common diseases. Therefore, the placenta and after birth, the mother's milk both help the baby to become strong. Right? So, this is the whole subject. Human reproduction is done. Now, after this, I will give you some questions, important questions already I have discussed, and go through them and At the end of that, tomorrow, next class, I'll discuss all the questions also. Then we will start with the next chapter, that is about the human reproductive health. Okay, done then. Just go through them, understand them, pause it. Once you observe, that's done for this class. Okay, see you.